Welcome to Unchatted Waters. In this episode, we'll be donning our life jackets, putting on our wellies, and exploring our scientific surveys. We'll discuss how we sample across the entire food web, from the smallest phytoplankton to the largest fin whales. During our voyage, we will talk about the people and technology that help us understand the ocean's ecosystem. So welcome on board. So welcome to Unchatted Waters, the podcast that talks to the people behind the science. I'm Kirsty, your host, and I'm so excited to introduce my two guests. Jeroen van der Kooij is a principal fisheries scientist, specialising pelagic ecosystems and focusing on distribution and abundance of pelagic species in relation to their physical, chemical and biological environment. Sam Barnett is a fisheries scientist leading in fisheries fieldwork. She's responsible for coordination of sampling teams during several fisheries and ecosystem surveys, both nationally and internationally. So welcome to the podcast.
Hi.
Hello. Great to have you.
Thanks for having us.
So I want to start with you, Sam, as a coordinator of some of these surveys. Can you describe what a survey is and why we need them?
Well, if we want to catch fish and we want to eat fish, we need to be able to keep a monitor on how many fish we catch and how many fish are in the sea. And to do that, we would monitor markets and what fishermen are actually catching and putting on the market. But then we also independently do our own fisheries surveys to monitor what fish are there in the sea. The impact of this is that we are able to sustainably fish and control what we're catching, making sure that we're not overfishing.
Perfect. So how have these changed over time?
Traditionally, surveys would probably target one particular species, the species that was of interest at that time. So that may have been something like plaice or cod. But over time, I suppose there was an appreciation that fish are not living in isolation. They live as part of a wider ecosystem, and you need to monitor all the components of that ecosystem. So on top of that, fisheries surveys are quite costly, and it makes a lot more sense to collect as many different variables from the different components of the ecosystem whilst you're out there. So it's that multi-pronged approach really that really helps us to understand A, what goes on, and B, do it for less money.
Can you give me an example of that, where that works?
One particular survey that I'm really interested in is one that targets the small pelagic fish community. Now, small pelagic fish are these silvery fish, often not very exotic looking, but they are extremely important to the ecosystem because they channel all the energy from the lower parts of the food web to the higher part of the food web. So they feed on plankton, for example, and they convert it into energy, and then they form the food for commercially important whitefish like cod, for dolphins, for whales, and for seabirds. So understanding what happens with those small pelagic fish is really, really important. When we designed the survey, why we focus not just on those little fish, but also on the other components of that ecosystem. Measuring the plankton, we're looking at the temperature, we are even counting birds and mammals, and in more recent years, bluefin tuna, which is one of the more recent additions to our ecosystem after having been away for many decades.
Amazing. So I'm imagining we're going out on a vessel and we're going out to sea and we're doing different samples and sampling. But what kind of vessels do we use, Sam?
So over the years we've had many research vessels, but we also use a number of different methods, including going on commercial boats and chartering commercial boats to use for fishing survey.
Yeah, if I can add something there, there's a distinction between collecting data on commercial boats during their routine operations and that sort of work we do on the research vessels. And that's primarily because whilst we can collect really useful information from commercial vessels, they tend to go to the areas where they know fish are. That's their job. So they're very good at that. And sometimes it's important, particularly when you want to monitor a stock, to also know where the fish are not. So a commercial fishing vessel would have no interest in going there. And that's why we go to all those areas with our research boats to look at what we call fisheries independent monitoring.
So does that include going to specific sites you've been on year, or is that kind of looking at where the fish might be in a larger area? Can you go into a bit of detail?
So it depends a little bit on the type of survey. A lot of the routine fisheries surveys that we do in the lab that have been conducted for many decades, they tend to go to the same say, 70-odd stations in the North Sea to look at the changes in fish abundance and distribution. But there are different designs, so you don't necessarily have to go back to the same area all the time, but you do generally look in the same wider area. But there's other types of surveys whereby you don't actually trawl at fixed stations, but you record acoustic data. And that's a tool that we use more when we study the small pelagic fish community. They're not always found in the same locations, as you can imagine. They move a lot more. So if you were just trawling at the same location year in, year out, you're very likely to either catch nothing or catch a very large school, and it would not necessarily help you in understanding what the trends are in that population. So a tool like acoustics, sonar, you may have heard of, or fish finders, means that you can sample much higher resolution. You have a much higher chance of encountering these schools over your total survey area, giving a much more accurate idea of the stocks.
So why are those historical data sets so valuable?
Well, they help us understand what happens over the longer term. So a lot of these fish stocks, they move, process is quite slow. So you may not necessarily see any changes from year to year, particularly with some of the demersal fish that are sexually mature after many years. Now, demersal fish, for those that are not aware, are fish that live on or near the seabed, basically. So cod is a good example. Other demersal or flatfish are things like plaice and sole. Now, they are sexually mature after several years, which means that the changes in the stock may not manifest themselves as quickly as in other species, which are sexually mature after a single year. So any change, for example, in sardine or herring may be much more apparent from year to year than that of some of the slower-lived species. So yeah, understanding those long-term trends is really important. It's very difficult to see patterns when you look very closely. It's often better to look at a larger scale, both in time and space.
So we've talked about some of the surveys, what they are and why we need them. But what is it like being on a survey? Sam, can you go on to a personal trip for us and share some of your memories and best bits of being at sea?
I mean, you have to be very aware that you're very privileged. The kind of things that you see at sea and the kind of things you do, although it's hard work. I mean, we joke about it being a holiday with fish. You know, you get to see some amazing things that people would love to see, like dolphins. You know, when you're standing at the front of the boat and watching dolphins bow ride, you know, that's an incredible thing to be able to just watch while you're working. There are challenges to it. The weather can be an interesting one. It's not always nice in the North Sea at certain times of the year, a lot of the year, to be honest. So, you know, you do get some very bouncy days. Seasickness is a big worry for a lot of people. Some people just don't get it. Some people do feel unwell and have to kind of manage that in whatever way they need to. Hours worked can also be a bit of a challenge as well. You know, we are working long hours on the boat. So, you know, it is a tough, grueling, sometimes 12-hour shift. But I think the positives just completely outweigh them whenever you go out there. So
what's your main highlight?
Main highlights are probably just the kind of species that you see while you're out there. I mean, from weird and wonderful fish, I mean, seeing things like octopus and sharks and rays, I mean, that's really cool. But also then seeing the seabirds. I mean, I really like taking photos, so there's a few of us that get quite excited about the seabirds and mammals on the boat. So we kind of, you know, you'll get some kind of shout or call from the bridge saying there's some weird and wonderful birds that you don't often see, and then we'll all want to go and see what that's all about. So yeah, the stuff you see out there is probably the main thing.
Yeah, and in the North Sea you can sometimes see the Northern Lights, can't you? Have you seen them?
Yeah, no, I haven't. I mean, we've tried so hard, like the times that we've been far enough up north to kind of really have a chance to see them. And I think we've just been really unlucky with it always being cloudy every single time we've had an opportunity.
There's a list in the bridge, isn't there, of everyone's name with the whole crew, just to let us know if it's there. But we haven't seen it either.
Yeah, no, it's still on the list. But orca this year were the big thing for me, I think, seeing them. So that was my little highlight so far this year.
Amazing. Jeroen, your turn. Give us your best sea-going story.
Well, that's a very good point, and I very much agree with Sam. It's that privilege of calling the sea your office for however long is the best thing. And I think from a scientific perspective, it's great to see all the different components that you're recording data from. So from the temperature to the observations on the echo sounder, like the schools that you may see, to the observations that the marine mammal observers may be making. It's being able to tease those pieces together and form why you're seeing what you're seeing. It's great to see. A particular example, I remember we were steaming off the North Cornish coast. It was actually quite a nice day, but there wasn't much happening acoustically on the echo sounder. When suddenly we saw on the thermosalinograph, which is a piece of kit which records the temperature and salinity of the sea surface, changed quite drastically, which suggested that we're entering a different water body, what you could call a front. And at the same time, we started to see very large fish schools on the echo sounder. And at that point, we had a call from the bridge saying that they started to see fin whales feeding at the surface and bluefin tuna joining them. So it's that sort of thing that gets everyone really excited. And plus, it helps us understand, the whereabouts of some of these organisms, including the fin whales. Why are they specifically there? Probably because there is a change in the water body. It drives local productivity, potentially increasing plankton, which then has a knock-on effect on the higher levels of the food web. It's a real privilege.
So you know why you're going out, but you never know what you're going to see, and that's the real draw, isn't it?
Absolutely. It's an adventure every time. Yeah.
Amazing. So Jeroen, you've spoken a bit there about fin whales, plankton. How do you measure the entire ecosystem from those two parts of the food web, from primary production with phytoplankton all the way to those large predators?
Well, that's a very good question, and there's not a single answer to that. A good start is to really put a lot of sensors in the water. There's equipment like the ferry box, which is a temperature and salinity measuring instrument, which records information wherever we steam. We have our echo sounder running all the time. That records all the organisms effectively in the water column at very high resolution and over very large ranges. We sometimes struggle.
So the jargon alarm has gone off there.
Okay.
Can you go over exactly what the echo sounder is doing? Because you've said it a couple of times. And I really want to tell our listeners how exactly it works.
Yes, no, absolutely. An echo sounder is a piece of equipment which is used not just by scientists, commercial fishermen, and anyone with a boat often. It's based on the concept that a pulse of sound is emitted from the hull of the vessel, from a transducer. It bounces off organisms and the seabed, comes back to the vessel and gets recorded. And by measuring the time it takes for the echo to return back to the ship, and also by measuring the intensity of the signal, you can establish what type of organism you're looking at and what depth the seabed is. So it's a really powerful tool. It's non-invasive, so you don't have to stick a trawl over the side. It's a bit comparable to flying over a rainforest and looking under the canopy, I suppose, something that you cannot achieve with any other piece of equipment. So it's very powerful.
Can you understand the abundance of fish from that intensity of the signal as well?
Yes, in principle that is possible. You need additional information and you need to make sure your system is calibrated. So you use a reference target to make sure that you understand the strength of the signal. By having that done and by collecting information about what species may be out there, because that's not always clear from an echo sounder on its own, you can definitely collect more information about the biomass of the organisms that are out there. So particularly for small pelagic fish, it's really the best tool.
The jargon alarm again has gone off for pelagic.
Oh, okay.
So I think you described what demersal was, but you haven't described what pelagic actually is.
Pelagic means open water in theory. So in principle, all the fish that are not associated with the seabed but more in the open waters are what we call pelagic. Small pelagic fish are often the type of fish like the herring and the sprat, the small silvery fish that form these very large aggregations. A tuna, for example, is also a pelagic fish, just a large one. The opposite of demersal.
So they're highly migratory. They're moving around a lot. They're not constrained by living in a rock pool, for example.
That's
they're out
right.
in the ocean,
Yeah.
exploring and seeing what they can find when
Exactly.
they're out at sea.
Exactly. Yeah, many of them are indeed, they're not associated with a particular seabed substrate like a lot of the demersal fish are. So they have much more freedom to roam. They still have a preferred habitat that's likely to be driven by temperature or food. They're much more versatile in their habitat occupancy. Adventurous,
Yeah.
that's what it is.
So thank you for explaining what an echo sounder is in a bit more detail. I was going to go to you, Sam, and ask, Jeroen mentioned things that need to be done in combination with an echo sounder. Can you go through a couple of examples of how we validate what we're seeing?
When we're out doing an acoustic survey and we're running acoustic transects, when we see a mark on the acoustic sounder that we want to look at or think is a fish or is a shoal of fish, we will then use trawls, fish on those marks to try authenticate what this these marks actually are, and if they are what we think they are. So over time of doing these trawls and looking at these marks, we've been able to kind of slowly get an idea of what we think they might be. So now we can look at a mark and say we think this might be pilchard, for example, but we still need to ground truth that and say yes, that is pilchard, we know that is. Or if we are not sure what this mark is, to be able to fish on it and say, well, actually this is herring, for example. So
ground truthing is validating or confirming what you've seen is correct.
Yes, yeah. We don't try and catch the whole school. We're just literally trying to kind of take a little scoop of what's there to get a relative idea of the whole community of fish that is within that school. So once we've caught those fish, we then go through a process of biological sampling, which involves sorting the fish into species. So if we've got a mix, we then go through different sampling techniques, including collecting the lengths, weights of the fish, sex and maturity of the fish. And then we also collect the otoliths, which are small little ear bones inside the fish's head that actually grow yearly like tree rings. So once we've collected those otoliths, we can then look down a microscope, and using the light from the microscope, we can see those rings very clearly and count them, and we can say whether a fish is 3 years old, 10 years old.
Brilliant. So is that the only kind of physical sampling you do? Is a trawl over the side of the boat?
Not necessarily. It kind of depends on what the survey is trying to achieve. Usually we will try and do as much as we can on these surveys. We try and get as much data as possible. And so usually the trawls go hand in hand with things like water sampling, which we would do to collect things like salinity. And then also we could do things like plankton sampling. So we put a different type of net over the side, which collects really small things. So the small plankton, zooplankton, which is the kind of little animals, and then phytoplankton, which is the plant life. Sometimes we'll analyse that on board. So we'll use microscopes on board to look at the zooplankton. We're trying to look sometimes for fish eggs and larvae. So we're looking to see whether the fish we're catching in the trawls are spawning, and if they are, whether there's eggs and larvae around as well. So that's something that we will look at on certain surveys.
So you've talked about plankton there. Can I just jump in and ask if there's any kind of automation to plankton sampling, like Jeroen mentioned with the acoustics? That's very non-invasive. Do we have anything similar for plankton?
The plankton imager for zooplankton sampling works by running a water system, a water flow from underneath the vessel through a camera and a field of view that will take images of everything that's in that water flow. It's running continuously as we're surveying, so it's great from the fact that we can get data basically from the moment we leave the port to the moment we come back into port. It's just running pretty much continuously. It can take photos of what's in that water column, so copepods, eggs, larvae, anything. So a copepod is a little bug-like thing that is one of the main sources of food for fish. It's really important, and there's multiple. So copepod isn't just one thing, there's multiple species of them. They are really important for small fish larvae as they're growing and developing. They will be eating these copepods. So large numbers of them are really important. You want to see large numbers of them. This PI will be able to take photos of these species that we can then run through artificial intelligence, which can identify them. And we also use, again, in a way of ground truthing, we will use taxonomists who will also look at those images and identify them to make sure that the AI is doing the right thing and identifying them correctly.
And for those of you who don't know, a larvae is a baby fish. So just imagine that egg hatching into a lovely baby cute fish with a little yolk sac on there. And we'll try and share some of the images on the infographic with this episode. So we've gone through a lot of information there, image and analysis of plankton. So you've talked about zooplankton. Is there anything for phytoplankton, Jeroen?
Yeah, there are a number of different tools out there. Probably the most conventional one is a fluorometer, and that records fluorescence. That can be converted into a proxy of chlorophyll, which is the pigment that's produced by phytoplankton. The amount of pigment in the water can be translated into the amount of phytoplankton. So I've just talked about fluorescence and the fluorometer, but there are other tools as well, like the flow cytometer, which is again is an automated sampling device which, put simply, measures the different size categories of plankton, which again is helpful in understanding the different functional groups of those very small planktonic organisms. That helps us understand the ecosystem a little bit better.
Wow. This is becoming such a good school day. I'm learning so much in this episode. Thank you so much. So we've gone through plankton, fish. So what about mammals? How do we observe mammals in the ecosystem? Who wants to jump in on that? Because you both talked about in your stories how much you enjoyed interacting with mammals. So opening the floor.
3, 2, 1, jump! Depending on the survey, depends on what we do regarding marine mammals. On our acoustic surveys, we usually have marine mammal observers. They come on board and they work really long hours standing on the bridge from pretty much the moment the sun's up to the moment it gets too dark to see anything. And they're looking for any cetacean activity, marine mammal activity, also bird life, so it's not just the mammals. And they have their own recording system to note down what species and the numbers and abundance of species that they're seeing as we're going along on survey. So they can keep a track of everything that they're seeing up on the bridge while we're doing other work around the vessel.
So is it safe to say these surveys are a big collaboration between lots of multidisciplinary scientists? Would you say that's fair?
Yeah, that's probably fair. These acoustic surveys are particularly suited to marine mammal and bird observers, which means that it's a lot easier for the observers to record useful information. By collecting this data on a number of different trips, including on different vessels from different nations, you get a large-scale coverage of the presence and abundance of these organisms. Yeah. And in addition to mammals and birds, since 2014, we are also starting to see Atlantic bluefin tuna.
So I've got a question to ask, because I think other people listening might have a similar question. So you've talked about echo sounders, and a lot of us might be aware that the way that mammals hunt for their food is very similar to sonar. Is there any interaction between the echo sounder and dolphins? Can you detect them on there?
Funnily enough, we can see dolphins on the echo sounder, not their physical presence necessarily, but we can see their clicks on our echo sounder. So the hydrophones that are part of our echo sounders that listen out to sound in the water pick up the sound of clicking dolphins, yes. But it's good to point out that the power and the frequency of the echo sounders that we use for scientific purposes are not strong enough to cause any distress to the dolphins, as far as we're aware.
You know when they're all around, because you just see these kind of like lines of like interference in the acoustic data, and you kind of usually then you look outside and they're all around you, and it's just like, oh, there they are.
They're here. Yeah. Okay, so we've talked about the lower parts of the food where plankton, zooplankton, fish, mammals, seabirds. Why is it so important to look at the whole ecosystem? You've talked about cost already, but why do we need to do it on such an ecosystem approach? Because it feels pretty intuitive that we need to do it that way. But why do we do it that way?
Well, that's a good question, Kirsty. The main reason, in addition to, of course, the cost that you already mentioned, the main reason that we monitor the whole ecosystem is that without understanding what drives the population size, we will never be able to predict how future changes in, say, the temperature or climate may affect the populations as we're monitoring them. So it's about getting that ecological understanding of the whole food web and all the drivers that influence it that we're trying to figure out. These ecosystem surveys, they monitor fish. That information feeds into the stock assessments, which help manage those stocks. Some of the variables that we collect are also used for monitoring the health of the ecosystem as well. So this may be nutrification, which is when there's too much nutrients in the water column. It may be the presence of particularly rare seabirds, particularly endangered Balearic shearwater that we've encountered on our surveys. Or it may be anecdotally monitoring a species like the Atlantic bluefin tuna. But collectively is where its powers of these surveys lie. Because it's only then that we start to understand our oceans and seas.
In terms of what our future surveys might look like, we've talked about so much technology that you use on these surveys, which are really valuable. How do you think that's going to change in the future?
I think sampling on board surveys is going to just change continuously. New ideas and new methods. For example, the PI, the plankton imager that I mentioned earlier, was collaboration between two organisations that wanted to try all this out on board, which is what we did, and it's now pretty much a permanent fixture on our surveys. So things like that, trialling out new stuff on board and getting new minds involved in what's going on board is how things are going to develop in the future, and encouraging things to develop in the future. One thing I've seen more of in the last few years is eDNA sampling. When I first started seeing it happen was students on board coming to look at eDNA, so environmental DNA, which is basically taking water samples when we were fishing and when we were seeing marine mammals to try and see whether they can pick up traces of those animals within the water. You can find the unique DNA patterns for that individual species, and you can say, yes, this is a mackerel that's been in this area based on the DNA pattern that you find within the water.
The next step will be to understand what the quantities of eDNA mean in terms of how much of that particular organism is out there. So certain times of the year you expect more DNA to be shed, for example during spawning periods. A larger quantity of DNA may not necessarily mean more fish, it just means that there's more secretions. So it's a very powerful tool. We're still, I suppose, in the early stages of it, but it's definitely something that in collaboration with students we've managed to develop.
Is there anything else we might use? I've heard of gliders, which are like little planes that go underwater, and can you describe some of those funky bits of technology which might be useful in the future?
Yeah, some of those, they're called gliders. Some of them are surface vehicles. They just operate from the sea surface, a bit like a boat, but then without anyone on it. It still needs someone to drive it from a distance normally. The point is you can get more of these smaller vehicles out there at lower costs. Other gliders that are being used are those that undulate through the water column. So they zigzag from the surface to the seabed and they zigzag through the water column. Those are often used to understand the oceanography of the water column. But of course there are other tools like remote sensing techniques which have been used for a little bit longer, like satellite. But the idea is that we always have to look ahead and see how we can make our data collection more innovative and cheaper as well. Plus the other advantage of some of the gliders is propelled by wave power, which means that we can be greener in the process of surveying the marine environment.
Wow, do you think you'll ever get to a stage where your echo sounders are released on buoys that can either stay put or drift around the ocean?
Well, yes, Kirsty, that we certainly can. In fact, we have already done this. So we've not used it in a traditional monitoring sense, but we've certainly released our echo sounders on surface vehicles and recorded the dynamics of the small pelagic fish community and the wider ecosystem.
We're getting to the end of our episode now, and I just wanted to go over a couple of fun facts. I don't know why I'm always talking about fish at parties, but it seems to be what I always do. So I refer to these fun facts as something you can take to a party and say, did you know I listened to this on a podcast? And did you know this cool fact about fish? So I want to go to each of you and for you to give us each a fun fact. We're each pointing at each other to go first. Yeah. So I'll say, Jeroen, you go first.
I don't think it comes as a surprise that my main favourite subject of conversation, maybe not at parties, but my main subject of conversation is the echo sounder. Because to me it is generally an exceptionally powerful tool. It does really record non-invasively the entire water column at centimetres resolution over hundreds of thousands of kilometres potentially. So I think that certainly deserves a mention. I'm not sure you get many laughs, but yes, it's a very powerful tool.
Conversation opener, it is. Very good.
Did you know?
Sam, to you.
The one that really fascinates people and being able to tell a fish's age the same way you count tree rings on a tree. So these tiny little ear bones inside a fish producing rings yearly, which you can literally just look at and count is pretty cool, I think. It's something
cool.
that not many people would even know about, that you can age a fish.
Yeah, there's chemicals that tell a whole story as
I was
well.
gonna
I think
say.
it was pretty fascinating what you spoke about earlier. Well, thank you so much for your time. It's been such a pleasure to speak to both of you, and thanks for being part of the podcast.
Thank you for having us.
You're welcome. Thank you for listening. This podcast was made by CFAS, the Centre for Environment, Fisheries and Aquaculture Science. To keep up to date with us, follow us on social media using @CEFASGovUK. We hope you can join us again next time.